Welcome everybody to this ESPN ERKNet webinar. My name is Albertine van Eerde. I'm a clinical geneticist in Utrecht. I'd like to remind you that your microphones are turned off, but if you want to ask questions, please do so and type them in the panel that you find in your computer screen, and I will read them out loud after Beata's presentation. So we turn to Beata. Dr. Beata Lipska is the head of the genetics clinic at Clinical University Center in Gdansk in Poland. She completed her medical degree in 2002 and her specializations in pediatrics in 2009 and clinical genetics in 2014 at the Clinical University Center in Gdansk. She received her PhD and habilitation in 2015 in genetics at the Department of Biology and Medical Genetics, also in Gdansk. She performed research scholarships at the Paris-Descartes University, University of Heidelberg, and the University of Nagoya and Baylor College of Medicine. Beata's main areas of expertise include hereditary kidney disorders, glomerulopathies, and KAKUT in particular, genomic disorders, genetics of pediatric cancer. She was an associate partner in the FP7 project Urinomics, and currently she is a member of the Molecular Diagnostics Task Force of the European Rare Kidney Disease Reference Network, ERKNet. She served as a member of the board of the Polish Society of Human Genetics and the WEMS section of clinical genetics. Beata has been a member of the ESCAPE network since 2010, and since 2012 she's the genetic coordinator of the PODONET registry. She performed and supervised more than 5,000 genetic tests for the patients from PODONET, 4C and ESCAPE studies. She's the author of several publications on genotype-phenotype correlations in hereditary podocytopathies. Beata, the floor is yours. I'm very much looking forward to your talk.
Well, thank you very much, and a very good afternoon to everybody. Hope it's not too windy up there. It turned out that it's now the wind in the Eastern Europe, so actually behind my windows, but the more warm welcome to everybody. It's going to be a very, very basic webinar today. So for those people who really want to get the fundamentals of what we are dealing today in genetics. To start with, let me remind you that chromosomal genetic disease is your daily bread, regardless what kind of patients you are treating, whether those are patients in pediatric period, whether those are adults, whether this is already a gynecology section and prenatal. You will always have patients that have some genetic factors predisposing to what you are dealing in everyday clinic. And please notice that depending on the severity of damage to your genes, the symptoms occur as early as in prenatal period, and this is why mostly chromosomal diseases are detected through prenatal screening. Single Mendelian diseases is this what we deal mostly in pediatrics, as you see here, the peak of incidence. But even you, people who are listening to this webinar, you are still not prone from not getting a genetic disease, either a monogenic, either chromosomal, hopefully not, or the multifactorial. So really in the clinical setting, you will always have a patient that has genetic disease, even if he's not born, if he's not having a congenital form of it. So just have it in mind. So how do we deal about all this? So definitely it is the role of primary caregivers, of nephrologists, of neurologists, etc., to order genetic testing. But please remember, the genetic testing is completely different from ordering CBC or image studies, etc., etc. First of all, as always, you should identify who's eligible for a genetic test. Then you should ask the question to yourself, but also to the patient. Do you want to know whether there is something genetic running in your family? If the patient is fully informed of what is the benefits of the genetic diagnosis and what can be the pitfalls, what can be the side effects, what additional findings there can be. A good example is the wrongly assignated parenthood, for instance. Then, if you and your client or your patient agree together that you both are really going into performing a genetic test, then you should decide what kind of test they should be offered, and then order this test in appropriate lab with appropriate quality certificates, etc., etc. Then, when the result comes back, it's not only that you should be able to read aloud what is there, but you should also know what are the next steps. So what should be the follow-up? And as you see, as we are climbing all the steps from the very beginning, from having the patient suspected of genetic disease to getting the patient having a genetic diagnosis, all those steps, you as the clinicians are the persons in charge of the patient. But there is this guy, a very rare specialty, as rare as rare diseases, called a geneticist, a medical genetist, a genetic counselor, depending on the country you are coming from. There are different systems in Europe how you do it. But yes, there is some help for you. And let me show you a difference, how we, the genetist, approach the patient, and what is the difference between our point of view and your point of view on the same patient. So for you, the patient is the one that has the disease. For me, and the person who's at risk of having mutation is already my patient. For you, the causative agent is this something that is occurring in the very patient. For me, he's born, it's in Germany. I'll discuss germline mutations in a moment. Then you are interested in the particular organ. So if it's a kidney disease, you need to make a kidney biopsy and make the diagnosis. For me, the DNA is all over, so it's enough for me to draw the blood, to draw this, to ask for a saliva sample, to make a buccal swab, and I will still reach the diagnosis. Then, for you, the intervention is all the medications that you order. It's the renal replacement therapy, etc., etc. For me, it's something that I refer to as the blah, blah, blah. So predictive risk counseling, talking to the patient, discussing the options, telling what this means for him, what is the burdens, what are the results of having a genetic disease. And at the very end, I will be referring to this throughout the seminar today, the family. Family are not those guys that are just bringing flowers and chocolate as a thank you for you taking care of the patient. For me, the family is also the patient. They share the risk status. Sometimes I need the family to get the diagnosis in my patient. And again, just bear in mind that family and genetics goes together. We definitely need to take care of multiple members, not only the single patient. So yes, to drive you through those steps that I'm just recalling here in the corner, starting from having this first glimpse, yes, let's perform a genetic diagnosis, and going up the ladder all the way to provide the post-test. There is this genetic consultant or genetic medical doctor who can help you. And it's not only that he will explain to the patient what is there, but he will mostly explain what there is the recurrence risk and what is the heredity burden in the family. And here I already am listing to you this 1, 2, 3, 4 different types of genetic testing that can be ordered. Usually, when you think about your patients, you are thinking about diagnostic tests. Does my patient has this or that disease? I'm ordering the test because I asked the question, are the symptoms caused by the mutation causing this hereditary syndrome? However, the second step later is asking, there is this something in the family, and maybe we should do some predictive testing for other family members who are yet in asymptomatic phase, or maybe in a very reduced, limited level of symptoms. And here, the issue of testing children comes to my attention, and definitely here you would like to refer to having a genetic counselor, because there's very strict ethnical and social and financial consideration when you want to test somebody who's minor, who cannot decide for himself. So it's always that you have to weigh the benefits over the side effects, especially if this is the diagnosis of hereditary disease, so the diagnosis of the disease for the life. This is somehow related also to assessing the reproductive risk, because it's not only nice, okay, I know what my patient has, but then the patient asks you, doctor, can I have children? Can I have healthy children? And how can we reassure that my children are healthy? And you see here those two other steps, so reproductive risk assessment and prenatal pre-implantation testing. All this should be discussed with the very patient that just had a diagnostic test. And because even if your primary interest as clinicians was just to make the diagnosis, the feedback to the patient should also address all those points. And to do so, I recommend that you just find where in your hospital, in your area, there's genetic consultant, and he will do this blah, blah, blah for you. So what we do, the genetist. So we do exactly the same job as you do. So we examine the patient, we examine the results of all the additional tests that he had. But there are those two particularities that I will now focus throughout my story. One of those is to take the exact family history and to be very precise on taking the pedigree. And in the second part of the talk, I will touch upon the molecular tests. So what is the pedigree? Pedigree, I call it the genetic alphabet. Those are the very basics that just having a short look on a piece of paper, when I have all those drawings as shown here on the picture, used in the right manner, and we are ready to know everything what is running in the family. I will not go through all those ideograms that are listed here. Just to remind you that male is a square and a female is a round, and if we don't know the sex, then we just use the diamond. And let me go directly to the first example here. And I would like you to wake you up a little bit. And now, could you think for 1 minute, what is the most likely mode of inheritance shown here on this pedigree? And we'll be making a voting moment. We cannot make the voting while looking on the slide, so please first think, and in a moment we'll start the answer sheet. Okay. You will be able to choose from 4 answers: autosomal dominant, autosomal recessive, X-linked dominant, and X-linked recessive. Those 4 Mendelian types of inheritance. And my question is, which is the most likely mode shown here? Let me give you still some seconds. Okay, can we have the poll? Okay, I think we can finish the poll already. Let's see the answers. And I would say quite good. Half of you got the right answer. This is an autosomal dominant inheritance shown on the slide. So let me go back to the slide. For those who missed the diagnosis, the most striking feature shown here is that affected patient has affected parent. Affected patient has affected parent. So you see that the disease occurs in every single generation. There are no skip generation, and this is typical for dominant inheritance. Some of you were trying to indicate that this is X-linked, or rather sex-related, because mostly males are affected on this pedigree. But just please remind you that a man passes to his sons Y chromosome. So this disease cannot be X-linked because the man passes Y chromosome. So then the answer here is straightforward, and this is an example of autosomal dominant inheritance. So that was quite easy. How about this one? Could you again think for a second on this pedigree? Looks more complicated, but I think you rehearsed already. Same answers. Autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive. Okay, can we start the poll? Okay, let's summarize this. Most of you answered that this is X-linked dominant, and only a few of you noticed the consanguinity in this family. Let's go back to the slide. Okay, can we close this? So if it was an X-linked dominant, and if this is the mutation on X chromosome, then he would pass this X to all his females. The mutated X would go to all his females. Also, in those generations, when you have girls here, you have girls here, because man passes X chromosome to all his daughters, again, this should pop up. However, please notice that this lady with this man are actually related. They are second-degree cousins. So most likely, this family is carrying an autosomal recessive allele in this branch. Then they marry a member of another family also having this recessive disorder, and then follows this pedigree. So just showing you those two examples, please notice that the reading the pedigrees is not so straightforward as it looks like, and sometimes you really have to think how the family is, where the alleles are coming from, in order to get this answer. So let's move to the second part of my talk already. So having said the alphabet is the pedigree, so let's now use the dictionary of the genetic language. So I already used the word mutation, heredity, etc., etc. So let's just go through definition of those. And let's start with the word that should not be used anymore, which is mutation. Since last at least 5 years, we, the geneticists, decided that the word mutation is misleading, because the word mutation means that somebody is mutated, is changed, but it does not tell you whether the change is pathogenic or benign, whether it's disease-causing or not. So in 2015, there was the criteria for assessment of variants, so changes in the sequence of DNA, were established. And although those criteria are rather straightforward, still the results presented are coming into 5 classes. And let me just explain to you what are those classes. So the class 5, or something that is called a definitely pathogenic variant, where the probability that this exactly causes the disease is over 99%, is a mutation, it's a variant that is well known, well established, when there are in vitro studies that confirm that is pathogenic. And you can use this results to make decision in clinical management. But more often in renal diseases, you'll be dealing with a likely pathogenic variant. You see here the probability that it will be it will be disease causing are pretty high, but they are not 100%. With geneticists decide that if you have a likely pathogenic variant, you should treat the patient as if having class 5, despite small risk of overtreatment, because usually those variants seem to be pathogenic, but only the number of data of in vitro studies verification is still pending, and we cannot confirm the status. Regrettably, 10%, up to 10% of the results that you will order will be giving back the information that a variant of unknown clinical significance, or so-called VUS, is noticed in your sample. And here, we don't know what to do with the patient. You shouldn't use this information for your clinical management, and you should just monitor the patient and stay in touch with the laboratory, asking whether the coming studies in vitro, etc., decide and reclassify this patient and move the decision into the benign or pathogenic side. Now, just the other important thing when you read the genetic result is that you read that your patient is a homozygote or a heterozygote for a given mutation. With homozygote meaning that both alleles are carrying the mutation, where the heterozygote is just carrying mutation on one allele. And depending on the disease, whether it's a dominant disease, heterozygous status is enough to make the diagnosis and be causative. Well, in recessive, you need to have both alleles deleted. When you look at in the classic studies of sequencing, you could see the heterozygote as having those two peaks here, while the homozygotes were always showing a single peak. Now, in the advent of next NGS, next generation sequencing, you can very precisely look on a single strands of DNA and check whether your mutation is present in every single copy of DNA, indicating homozygous status, or whether it's present only in half of those as shown here. And in that way, it means that it's heterozygote. So these two concepts are already visualized from the genetic study. But the question arises when two mutations are found in the patient. As shown here, probably I hope you can notice the green mutation here present in roughly 50% of DNA sequences, and another mutation shown here also present in 50%. Now, the question is whether those two mutations are present each on a single allele, making the patient being a compound heterozygote, so having an allele inherited from one parent, a carrier, and having the second allele inherited from the second parent, also a carrier, and being causative for the disease, or whether this result indicates that two alleles are coming from a single parent, and in that way, both the parent and the patient is just a carrier of one damaged copy of the gene, even if there are two mutations. So in the old days, when you were receiving a genetic result, and some labs are still doing this, they ask you before issuing the final result, they ask you for blood samples of your parents, so that you can determine by genetic testing of them whether they carry both mutations. Nowadays, with NGS, if the mutations are close enough, we are lucky to see this already in a single test, and this verification is no longer needed. But please notice that when you read your genetic result, it should be explicitly stated whether you need to double check this through parents or not, because this changes completely the diagnosis. Here you have an affected patient, here you have just a carriership status. It's not so simple, and because we are talking about genetic language, so just on the result, it either written homozygote, or either it's written, this is the accordance to our genetic nomenclature, this is how you write that both alleles are carrying the same mutation. For compound heterozygote, you can have two different wordings. The upper wording suggests that two mutations were found, but we don't know where they're coming from. while the lower sentence says that we know that they are coming from the separate alleles. As you see, the square brackets and the haphalon here indicates that those two are confirmed to be on the both alleles. For the heterozygote, however, we just use the equation sign, which indicates that the second allele is clear, has no other variant. So all those writings you can find on the results of your patient, and you should be very cautious to notice whether you need further testing before issuing this result to your patient or not. I'm seeing I'm getting a little bit short on time, so just a few words on penetrance and expressivity. Penetrance, when you read about any single disease, a genetic disease, you will always have in the very first sentence the percentage assessing penetrance over different features and also the expressivity. Penetrance tells you what percentage of patients with the mutations have symptoms, okay? While the expressivity tells you to what extent a given feature is present. So a good example here would be a tall statue in Marfan syndrome. All Marfan patients are tall, but to what extent, how tall they are, depends. So you see, the penetrance is 100%, but expressivity varies. And usually in autosomal dominant diseases, you will have both penetrance and expressivity as the describing features of the disorder. The other thing which is also important to bear in mind is the heterogeneity, which can be either the locus, as shown here for autosomal dominant polycystic kidney disease, which is caused by PKD1, PKD2, and as you know, a few more genes recently. So two different genes located in different chromosomes at the end cause the very same disease. While the allele heterogeneity, and I'm using the very same example of PKD, is when different types of mutations, so different variants, are either severe or more or less benign in the cause. So those 3 patients are diagnosed with the very same disease, but looking precisely on the genetic test will tell you whether the cause of the disease will be severe or not. So as you see, those 2 statements are also important to describe, to give back the patient information what will be the clinical cause in his particular setting. And the last thing that I would like to touch upon is the mosaicism. Mosaicism is the situation when there are 2 or more cell lines, so cell with and without the variation in the person. It can be constitutional, so the patient is already born having 2 or more lines, or it can be confined to the germline, so to your gonads, either to ovaries or the testes. And usually when you take the family history is that there is completely healthy parents. As you see, this man has 2 partners, and he was tested and his partners were tested negative for the mutation, but still from those 2 different relations, affected children were born. Probably one of his testes, there is a fraction of sperm cells that bring the mutation as the result of germinal mosaicism. However, the question is, we are all actually human beings, we are mosaic. Because if you think here that you are the zygote, being just 1 cell that you divide into 2, 4, 8, 16, etc., etc., and at the very end here you are the adult. And if we presume that this is the first cell that will made up the kidney, okay, and then you developed polycystic kidney disease because all the cells will grow with the mutation causing PKD. But then the patient comes to the clinic and says, oh, that looks like a PKD, let's take his blood. And this is the first cell of his blood. So we are drawing his blood and we find that he has no mutation. Can we rule out the diagnosis in him? No, because we look at the kidney phenotype and we are still consistent with the disease. So as you see, sometimes we need to test more than one tissue just to be sure that we are not missing mosaicism as the cause of a negative genetic result. So there is a lot of concepts in genetics that you should pay attention when reading a good genetic result from the laboratory. Usually those tests, they are few pages, and really it is important not only to read what is in the on the first page, but to go through all those lines and understand that not only a negative result does not rule out the diagnosis of genetic disorder, and there might be this mosaicism as the underlying cause of non-detection or the wrong technique used for searching for this mutation, but also having a patient diagnosed with the disease due to locus and allele heterogeneity, due to variable expressivity, non-full penetrance, the two patients having the very same mutation in the very same gene may still have completely different cause of the disease. I'm not touching upon here the modifying genes, and I'm not touching upon environmental factors, but there is more and more complexities related just to the single test that is drawing some blood for EDTA and having a genetic test. So just as a summary note for today, please bear in mind that it's although it's so useful and so handy to have genetic diagnosis in your patient, a lot of blah blah blah, a lot of messages that come with this diagnosis should be transmitted to your patient, should be discussed in multidisciplinary setting, and that there are genetic counselors who will spend the time at explaining the heredity cases to your patients and will explain all those homozygosity, heterozygosity confirmations before the patient can make autonomous decision about reproductive issues, testing other family members, etc., etc. So I hope I just presented at least a few basic concepts that that will shed some light on the language that geneticists use to describe patients with genetic diseases. And I will stop here. I'll take your questions. And of course, I'm already advertising for the coming webinars listed here on the slide. Thank you very much for your attention.
Thank you very much, Beata, for this very nice overview of basic genetics. I really liked the idea of alphabets and dictionaries, and the metaphor that came up for Beata is maybe Beata and the genetic counselors in your own hospital, you might see them as your librarians. So ask them for help if you don't understand something, in order to have better patient care. So I see there's not that many questions. There's April Yilmaz saying thanks, so that's extremely nice, but not a question. So I would really urge you all, if you'd like, you can ask your questions. The webinar will be recorded, so you can also hear it back later. I myself have a question for Beata regarding the predictive testing in Poland. In my country, it's the predictive testing is kind of the domain of the clinical geneticist, so the other doctors don't or shouldn't do it. How is that in Poland?
Yes, I mean, thank you very much for paying attention to this fact. Yes, predictive testing, if you have a completely asymptomatic patient, that should be definitely performed only after genetic consult. So then it's ordered through the geneticists also. And I think this is the official statement of European Society of Human Genetics. If you want to do predictive testing, you do it through genetic consults. The exception to this rule is oncology, when you have the family history of cancer, because there are so many people having oncology history. that there would be not enough counselors for that. But in internal medicine, this is done through geneticists, because we do the blah blah blah. We just wage the benefits against the side effects, especially in children.
Yeah. I think this is an important point. Still, there are no. Oh, there's a question. Menka Yadav. Sorry, I need to read out loud. Hi, thank you for a great talk. Just wanted to ask regarding the concept of pseudogenes and their implications on exome analysis. So that's a pretty detailed question for a basic talk, but nevertheless, I think it's important. So could you maybe comment on that?
Yes, indeed, especially that I was talking about PKD1, which is a very well-known gene for pseudogenes. So I didn't touch upon that, but if you are ordering a genetic test for your patient, you should be very, very careful to choose a good laboratory for that. And you should be sure that the technique the laboratory is applying is really covering the difficulties in testing the gene and addressing this. Because different diseases have different requirements. I didn't go into detail, but pseudogenes are not as are the genes that look almost like the gene of the causative for the disease, but still during the evolution were kind of switched off, and they make the bias for the reading through the automatic reading sequence in whole exome sequencing. So for those genes, you should be very careful and have a more precise tool to address this issue. So yes, whole exome sequencing can miss diagnosis there.
Thank you. So no other questions. Yeah. Sorry.
Albertine, you of course could add on from your point of view.
Well, I think basically the post-it note on your slides says it all. You really should take care to understand what was tested and what could have been missed in many perspectives. And I think a multidisciplinary environment for that is extremely important. I was wondering, there's no other questions online right now, although it wasn't part of your talk and it will be part of other webinars. But for this basic, for these basic concepts, could you maybe touch upon briefly which domains in nephrogenetics are more suited than others for a genetics first approach?
Okay, it would be easier to answer on the reverse. If you have acute, if you have isolated congenital defect of kidney, the chances that you will find an underlying genetic cause. For isolated cases without intellectual disabilities and problems from other organs, here the mutation yield is low, while for tubulopathies and glomerulopathies, the mutation yield is much higher and it's very handy in a clinical setting. So I would not think about genetics in isolated CACUT. I would think in all the other fields of nephrogenetics.
Yeah, maybe unless there's a family history.
Exactly.
I might probably add, yeah. So now there are questions. So Simone Baldovino is asking, thank you for the excellent talk. Can you explain more about lionization for X-linked diseases?
Okay, lionization means that one of your, in females, one of your X chromosomes is switched off, and it is switched off randomly. So we never know which X chromosome is switched in which single cell. That occurs very early in the embryological development. It's estimated around 10 weeks of gestation when you already have your one copy switched off. And you can be the lucky one to have most of your mutated ones switched off. And then your phenotype is rather benign because the other copy of X chromosome is working fine. or you can be unlucky and most of your mutated chromosomes, X chromosomes, will be remain active, producing the broken protein and hence producing symptoms. A very nice example of lionization as the factor of clinical presentation is Alport syndrome, which is X-linked dominant. And here we know that females can have a wide range of spectrum, usually much more benign than men, but still those few that have it more severe, maybe due to bad luck in switching the wrong copy of the X chromosome. This, however, cannot be easily tested because, as I said, it was which X chromosome is switched on is done already at the 10 weeks of gestation. So those single cells are already know that they have to switch it on, and you cannot test it. The test in the blood is not corresponding to the test performed on the cells from your kidney, etc., etc. So you cannot estimate this. You just estimate this from the clinical manifestation of the patient.
Okay, thank you. Maybe Simone can type more if he needs more explanation. I thought this was this made it rather clear. So there's another question from Kenneth Lieberman. He asks, could you elaborate on the process of deciding the likelihood of pathogenicity for a new variant? And in particular, he says, how do you use population frequency versus in silico protein structure estimates? And maybe this goes back to your ACMG slide.
Exactly. I mean, that's a question that I could answer for at least 1 hour, and we don't have it.
No.
So let me just go here. I hope you can see this. So ACMG criteria say that you should use a lot of different criteria, and the frequency of population is one of the important things, but it's not determinant because you know that we have the founder effects. If you would use the threshold of less than 1% population, we would have issues with cystic fibrosis, thalassemies, etc., etc. Also, please remember that the databases we are using are mostly based in United States. There's also a good database in United Kingdom, but they're not such good databases for example, for Asian population or for Eastern Europe populations. So something that you think it's extremely a rare variant may turn out to be completely frequent benign polymorphism in those countries that are just underrepresented in databases. On the other hand, in silico predictors are only in silico predictors. They are based again on the amount of data we have on the structure of the protein and its conservation. And if we are talking about proteins that are newly discovered, then we don't have this information for those proteins yet. And then in silico predictors are useless. So if you go back to ACMG criteria, you will learn that there's, I think, 15 different factors that score from 1 to 3 in strength, and only adding up to all those points make you final decision. So you should be very, very careful with that. You should, if you don't agree with the genetic result, you should go back to the lab and discuss this. Also, some mutations published back in 2000s turn out now to be false positive, and there is some recent journals that recall all this previously known pathogenic mutation actually is a benign one. So updating this information, going through, and always having the patient with her phenotype in front of you as the final determinant of the phenotype, rather than what is in the ACGG sequence of his genes.
Yes, thank you. There is another question by the same colleague who previously asked about pseudogenes. Menka Yadav likes to know, do mitochondrial genes, so the not nuclear encoded mitochondrial genes, get covered with exome sequencing? Do we need to do some special test or method? ? Okay,
it's a very good question. It depends on the laboratory you are using. As the general rule, whole exome sequencing is not covering mitochondrial genome. However, good laboratories add on to their whole exome information on mitochondrial genomes also. So they advertise, oh yeah, better because we also screen mitochondrial. And you can do it in a single test. You just have to be aware that that it's available in your lab. Also, we didn't touch upon this, that exome tests for exons, so for coding proteins, but there is a lot of regulatory sequences and deep intronic variants that can also be pathogenic. And usually, good laboratories also enrich their either panels or whole exome sequencing and add information on known in regulatory elements also. So sometimes when you see the advertisement, you will get this information ready there, that not only exonic sequences, but also intronic and also mitochondrial. But it's not the rule that is there. And there's no official standard minimal set of genes that should be tested. So you cannot sue your laboratory that overlooked that or not, because there are no standards yet as officially. So you can just try to find the best offer available through your insurance system.
Thank you. No questions came in anymore, so I'll talk a bit more and see if somebody's typing, but right now, no. Tanya, who's here on behalf of ERKNet and organizing everything wonderfully, keep an eye out on the typing. So I'll start reading the closing lines. Beata, first of all, thank you very much for this very nice presentation again. I'd like to thank the audience for your attendance, and you can look this webinar. You can look it up if you want to check something out. By this, and no questions came in the meantime, I would very much like to close the webinar and see you next time. Bye bye.
Thank you very much and have a good evening.
Bye bye. Thank you.